Okay, so in this video, we are going to talk about the white blood cells or leukocytes and platelets. Okay, so what are their functions? What are the pathological conditions associated with them? So white blood cells, again, they include different types of white blood cells. For example, neutrophils, monocytes, eosinophils, lymphocytes, and basophils. Okay, these are the major differential white cells or leukocytes present in them. And as we looked at in the previous slides, for hematopoiesis, the lymphoid cells will produce T lymphocytes and B lymphocytes. Okay, so they are important. And the myeloid cells, the myeloid stem cell will produce all the remaining cells, eosinophil, basophil, monocytes, and neutrophils. So these are collectively called as white blood cells. Okay, so first we are going to talk about neutrophils. So what are neutrophils? So we know that these are the most abundant type of white blood cells present in the body. Okay, around 50 to 70% of the cells will be neutrophils out of white blood cells in the body. They are approximately twice the size of red blood cells. So red blood cells we said around 10 micron in size. So this will be around 20 micron in size. They will have a multi-lobed nucleus or polymorphonuclear cells. You must remember this word. Polymorphonuclear cells or multi-lobed nucleus. So if you look at their nucleus, they will have a multi-lobes. Okay, it's like a small lumps. So they will have multi-lobes. So they are called multi-lobed nucleus or polymorphonuclear cells. And they do have a granulated cytoplasm that stains with both basic and acidic dyes. So you can see here in the cytoplasm, they have lots of granules. And these granules got lots of different molecules, particularly lysosome enzymes, in order to capture the invading pathogens and lyse them. Okay, so when you stain under the microscope, so you can stain these cells with both basic dyes and acidic dyes. That's why they are called neutrophils, which means they can accept both acidic dyes and basic dyes. Okay, how many cells are there? So it's around 2.5 to 7.5 times 10 to the 9 cells per liter of blood. And it's the largest population of leukocytes, I said before. And lifespan of these cells will be around 6 hours to few days. And what's the main predominant function of this fly is phagocytosis. So they will mainly engulf the foreign materials coming into the blood, particularly bacteria. When you get a bacterial infection, these cells will play vigorous roles in order to completely engulf the bacteria and get rid of them. So therefore, they are playing vital role in protection of the body from bacterial infections. So that's about neutrophils. Next, eosinophils. Okay, so what are eosinophils? They are approximately same size as neutrophils, and they do have a bilobed nucleus. Neutrophils had polymorphonuclei, or excuse me, multi-lobed nucleus, but these ones are bilobed. So you can see here they got only 2 lobes. Okay, so you need to remember eosinophils got bilobed nucleus. Again, they do have a granulated cytoplasm. They have granules, but these granules will stain only with acidic dye, for example, eosin red. Okay, that's why they are called eosinophil. So when you stain them with eosin red, these granules will become red color. Okay, that's why they are called eosinophils. They have large pink staining granules, and these granules contain histamine, plasminogen, DNAs, RNAs, and other molecules in them. So how many cells are there? It's around 0.04 to 0.44 times 10 to the 9 cells per liter of blood. They are really small population. We talked about before, 1 to 4% of the cells. So what's the lifespan of these cells? It will be around 8 to 12 days. Again, they are going to do the phagocytic function. So the main functions are providing immunoprotection and phagocytosis of antibody-coated pathogen. So if there is a bacteria or virus, and if the antibody is stuck to them, then they will remove them. They can also attack parasites. It's a specific feature for eosinophils, and they can cause allergic reactions because they got lots of histamines in their granules. So when they release the granules, histamine will get released, and it can cause allergic reactions. So these are the characteristics and the functions of eosinophils. Next ones are basophils. So what are basophils? They are small, slightly smaller than neutrophils, and they got a single lobed nucleus. You can see here it's got only single lobed. So that's why I'm saying you need to remember the shape of nuclei in these different types of cells. The neutrophils will have a multi-lobed nucleus, eosinophil will have a bilobed nucleus, and the basophils will have single lobed nucleus. And they do have a granules in their cytoplasm. They are heavy, and these cytoplasm granules will stain with basic dye called methylene blue. Okay, so when you stain with methylene blue, they will form large purple staining granules, and that's why they are called basophils. So neutrophils can be stained with both acidic dyes and basic dyes. Eosinophils only acidic dye, for example eosin red, and basophils can only be stained with basic dyes, for example methylene blue. So when you stain them with methylene blue, you will see large purple stained granules. Again, these granules contain histamines, heparin, and proteolytic enzymes. Their population is very low, less than 1% in the population of white blood cells. So they will have around 0.01 to 0.1 times 10 to the 9 cells per liter of blood, not microliter. Try to remember. They are not phagocytic cells compared to neutrophils and eosinophils, and the lifespan of these cells will be from few hours to few days. So what is the main function? They release histamine during inflammation. Again, that can cause allergic reactions. Okay, but remember, they are not phagocytic cells, so they can't engulf the cells directly. So the next type of white blood cells is called as monocytes. Okay, and these cells will become macrophages in the tissues. So when they are present in the blood, they are called as monocytes, and when necessary, they will get trapped into the tissues and they will transform into macrophages. So the macrophages normally will not circulate in the blood. Only monocytes will be circulating in the blood. For example, if you got an infection or if you got an inflammation, for example, atherosclerosis, the monocytes will go to the target site, transform themselves to become a large phagocytic cell, and that's what is called macrophage. Macro means big, and phage means phagocytic cells, which means it's going to engulf the foreign materials or debris of damaged cells. So they have large kidney-shaped nucleus, and they are larger than neutrophil, and it contains large population of lysosomes because the main job of these cells is phagocytosis. So they're going to eat something and then digest everything. So they should have lysosomes because lysosomes contain all hydrolytic enzymes that's going to chop everything. So the population of these cells will be very low, around 0.2 to 0.8 times 10 to the 9 cells per liter of blood. The lifespan of these cells will be several months because they will circulate until they get a job, but therefore they can circulate for a long time. So when they get activated in the tissues, they become macrophages. Macrophages will engulf the foreign and damaged materials. So again, it plays a vital role in protection from infection, and it will eat the bacteria, red blood cells, dead cells, and the cellular debris. So the main role here is phagocytosis. So the next ones are lymphocytes. So lymphocytes are important cells because that's the second most popular cells in the white blood cells. So they have a large and relatively round nucleus that completely fills the cytoplasm. Okay, it's a very large cells, and they are slightly smaller than the neutrophils, and they are around 1.5 to 3.5 times 10 to the 9 cells per liter of blood. Their lifespan, it can persist for many years. For example, memory cells, because that can carry the memory of that particular antigen or viral particle or bacteria that you encountered until it produces the antibodies against. They are divided into two categories, for example, T lymphocytes and B lymphocytes. And the T cells are important in direct immunity, and the B cells are important to produce antibodies. Okay, so their main function again in immunity. They play central role in the immune system, protecting us from infection, especially from viral infection. Particularly T lymphocytes play major roles during viral infections. And T lymphocytes can attack pathogen directly, and B lymphocytes will produce antibody. So these are the major functions of lymphocytes. Two different types of lymphocytes, T and B. T will produce main direct immunity, particularly against viruses and virus infected cells. B lymphocytes will produce antibodies, and those antibodies will attack the invading pathogens. Okay, the count in here, the neutrophils will get elevated in bacterial infection, stress, exercise, and myeloproliferative diseases. Lymphocytes will get elevated during viral infection and some lymphoproliferative diseases. Monocyte level will get elevated in infection, inflammation, tissue damage, and monocytic leukemia. Eosinophil will get elevated during allergy and intestinal parasites because that's a specific feature for eosinophils, and during myeloproliferative diseases. Basophils will get increased in myeloproliferative diseases as well. Okay, what are the pathological conditions associated with white blood cells or leukocytes? First one is leukemia, which is a really important condition, which is normally called as blood cancer. Okay, so what is leukemia? So leukemia is basically increased levels of leukocytes. So in our blood, if we have more than what is required or what is the normal level of white blood cells, then it's called leukemia. So this is called as a cancer of white blood cells, or generally people call it as a blood cancer. This is basically uncontrolled growth of one particular type of WBC that occurs in the bone marrow and causing a rise in cell count, obviously in the affected WBC type. It could be, for example, neutrophil, or it could be monocytes. It could be any type of white blood cells increased level. There are four main classes of leukemia here. So the first one here is acute myeloid leukemia. That's when we will see the myeloblast in the blood. So the myeloblast should not be in the blood. It should be only in the bone marrow, normal condition. If it's encountering into the blood in large quantity, that's called acute myeloid leukemia. Next one is acute lymphoblastic leukemia, which means the lymphoblast. Okay, it's coming from lymphoid progenitor. If these cells encountering into the blood or entering into the blood, then that condition is called acute lymphoblastic leukemia. Again, these cells must not be in the blood under normal situation because they should be only in the bone marrow to produce the cells. So acute leukemia, there will be little or no maturation, and is defined as the presence of over 20% of blast cells in blood or bone marrow at the clinical presentation. Then the next classes are chronic leukemia. So here, for example, chronic myeloid leukemia. So all these cells, for example, mast cells, basophils, neutrophils, eosinophils, if they are increased in large quantity, that's called chronic leukemia. So it will have many maturing or mature white blood cells. Okay, you need to remember here, acute leukemia means the progenitor cells or the progenitor cells or stem cells will enter into the blood. That's called acute leukemia. It could be acute myeloid leukemia or it could be acute lymphoblastic leukemia. Chronic leukemia means the actual mature white blood cells, their level is increasing. For example, chronic myeloid leukemia, where these cells or any one of those types can be increased. And the next one is chronic lymphoid leukemia, which means the B lymphocytes and the T lymphocytes can get elevated. So acute leukemia, chronic leukemia, you need to remember them. Okay, what will happen if the white blood cell level is going down? That condition is called leukopenia. Okay, it's basically reduced level of leukocytes. So it's a reduction in a particular type of white blood cell, or it could be a reduction in all types of white blood cells. It's mainly due to the bone marrow failure. Okay, if there's any problem in the bone marrow, if that's not producing sufficient number of cells, then automatically it will go down. So what are the causes here? So microbial infection, many viral infection that can attack, excuse me, it can attack the bone marrow, that can lead to this reduced level of cells, and then chemotherapy, radiotherapy, and other medications that normally people receive that can mainly affect bone marrow and therefore the cell production. Immunosuppressive drugs, they have adverse effects on the cell production, and then sepsis, which is basically infection all around the body. Okay, this is when complete collapse of immune system will occur. Again, that can lead to leukopenia. So these are about the white blood cells. Now we are going to talk about the last type of cells called platelets. The platelets are also called as thrombocytes, because they are important for blood clotting or thrombosis. That's why they are called thrombocytes. So again, so my area within cardiovascular, so I research mainly on platelets. That's why I'm a professor in cardiovascular pharmacology. Okay, we do lots and lots of research in platelets. So these are our images. You can see published in different journal covers. So this is how the resting platelets will look like. It's a small disc-shaped structure. Okay, it will not have a biconcave similar to white blood cells. So normally it will be spherical in shape. This is how it will look like, even though this platelet slightly got activated. So it looks like a snail. But when they get activated, they will make lots of finger-like projections, and it will become like a large film, like a plastic film-like structure in order to adhere to other cells to make a blood clot. So that's what we are going to talk here. Okay, so they are small anucleated cellular fragments of large precursor cells called megakaryocytes. So we've seen this before. Megakaryocytes, it's a large cell with a large nucleus. That's what is going to produce platelets, and then platelets will enter into the bloodstream. Okay, so platelets do not have nuclei because they will have all the proteins necessary for their function, and they acquire everything from their parental cells called megakaryocytes. Again, it's produced in the bone marrow. They are the smallest cells in the blood. They will be around 2 to 4 micron in diameter in resting condition. So if we measure this, it will be around 2 to 4 micron. But when they get activated, they can go up to 10 to 12 micron in size. Okay, they will appear in blood films as dark staining granules, and we have around 150,000 to 400,000 cells per microliter of blood, or 150 to 400 times 10 to the 9 cells per liter of blood. Lifespan of platelets will be around 5 to 10 days. The main role of platelets will be blood clotting. That's also called hemostasis. So when you get an injury, the blood has to clot in order to prevent the excessive bleeding. So that's the main role of these cells. So again, if you look at them under electron microscope, the resting platelets will look like that, even though some of the platelets got slightly activated. So these cells should not have any finger-like projections when they are not activated. When they get activated, this is how it will become like lots of finger-like projections, and then slowly it will become like a thin structure, like a plastic-like structure. So if you look at the blood clot under the electron microscope, this is how it will look like. It will have bunches of activated platelets stuck to each other. Then you got fibrinopeptides. Excuse me. Fibrinopeptides that's coming from fibrinogen. We talked about fibrinogen. It's a large protein, a large fibrillar protein present in the blood, and it's really important for blood clotting. So it will cover all the activated platelets, and automatically it will entrap lots of red cells and white cells as well within the blood clot. So that's how the blood clot will look like. So what are the different stages of platelet activation? So don't worry too much about the platelet activation in this lecture. I'll just skim through because we have the next lecture on blood clotting. So where I will talk about the blood clotting in more detail. So there are 4 stages of blood clotting here. Okay, so we will talk about the platelet activation particularly. So the platelets, because they are very small in size, they will be pushed towards the edges of the blood vessel. So this is the lumen of the blood vessel. So when the blood is flowing, all the red cells and the white cells will flow in the middle. The platelets, because they are really tiny, they will be pushed towards the edges. So in the blood vessels, we have single layer of endothelial cells. Okay, they are really important to cover all the connective tissues, particularly subendothelial collagen. So when you get damaged, this endothelium will get damaged. When it's get damaged, it will expose the subendothelial collagen. So that's the first stage in the platelet activation. When the collagen is exposed, collagen is an important activator for platelets. So the platelets circulating around these regions will see the collagen. As soon as they see the collagen, they will get activated. When they get activated, they will make a simple monolayer, single layer of activated platelets. These activated platelets will release lots of factors from their granules and from their surface. And these factors will activate more and more platelets and attract them. And finally, all the platelets will get stuck together in order to make a platelet plug or a platelet aggregate, and that will seal the area. In parallel, The blood clotting cascade will also be going on the surface of platelets. At the end of the cascade, we will produce the fibrinopeptides from fibrinogen. We will produce fibrinopeptide. So everything will get clogged here in order to make insoluble blood clot. Again, as I said, we will go through this in more detail in the blood clot lecture. So what are the pathological conditions relating to platelets? So the first most important one is thrombosis, which is basically blood clotting in pathological conditions. So during normal conditions, during hemostasis, if we get an injury, the blood must clot in order to prevent the excessive blood clot. So that's a normal blood clotting, physiological blood clotting. But if the same blood clot is happening within the blood vessel during disease conditions, that will stop the blood flow to main organs, for example, heart and brain. So if this is happening in the coronary arteries, it will cause heart attack within the heart. And then if it's stopping in the blood flow in carotid arteries, then it will stop the blood flow to brain. Then it will lead to stroke. So this is mainly caused by the rupture of atherosclerotic plaque. It's a lipid deposition. When it's breaking down, it will cause the blood to clot within the blood vessel. OK, so again, we will study in more detail about this in next year. So they are responsible for thrombotic diseases, mainly heart attack and stroke. OK, what if the platelet level is going down? That condition is called thrombocytopenia. It's a reduced level of platelets, and it can be due to any immune disorders similar to white blood cells, and it can be caused by certain mediators, for example, immunosuppressive mediators or immunosuppressive drugs. It can lead to uncontrolled internal and external bleeding because the platelets are critical for blood clotting. If you don't have enough platelets in the body, then we will bleed for a long time. It could be internal or external. OK, what is thrombocythemia? OK, this is a condition when we have more number of platelets than the normal level. So this is due to abnormal production in the bone marrow. And again, it could be due to tumors or cancers in the bone marrow that will lead to production of large amount of platelets, or other pathological condition that can also lead to the production of increased level of platelets. So it can cause accelerated blood clotting within the circulation. So similar to thrombosis, same thing happen here. If you got lots of platelets and if they are touching each other, they can aggregate, they can get activated and they can aggregate and they can make blood clot within the body, and that can cause problem. OK, essential thrombocythemia is a rare condition and it's a rare type of blood cancer because it's increased production of blood cells. OK, finally, destruction of blood cells. So we talked about the lifespan of each and different type, each type of blood cell. So after that lifespan, what will happen to them? I briefly mentioned before, these cells will go to spleen and the spleen will destroy all the wall cells. That's the main function of spleen. OK, it's an organ present in our body. So the spleen is an ovoid shaped organ found in the upper left abdominal cavity. It's around 5 to 13 centimeter in size. OK, and this is acting as a largest filter of the blood, and by filtering the blood, it will remove all the old and the damaged cells, and it will be engulfed by phagocytes. OK, it will have lots of phagocytes and it will engulf all the old cells and the damaged cells. This is how they are removing all the old cells, and the new cells are continuously produced by the bone marrow and they will be continuously replaced in the blood. So the old cells are one side getting cleared and the other side new cells are coming. So this is really critical to maintain our normal body function within the blood. OK, so these are the ones mainly happening within the blood. So in the summary of this lecture, so we talked about the role of each and every component of the blood, liquid components and also the cellular component. And we talked about how the blood cells are being formed, particularly hematopoiesis. And we talked about the structure, function and major characteristics. These are hematology of different cell types, including RBC, WBC and the different subsets of WBC and platelets. Then we talked about the pathologies, disease conditions associated with the different types of the blood cells. And then we talked about destruction of blood cells in the spleen. So this is about this lecture. So I listed some books here. So you can look at these books and go through this lecture and the screencast. And as I said at the beginning, please let me know if you have any doubts on this lecture. Thank you so much for listening. I look forward to see you guys at some point. Thank you.